Yeah, it's really a pleasure to be here with you all today. Yeah, I didn't realize that there was a phage focus. I would have been happy to talk about the phage-related work that we're doing in the lab. But instead, I am going to tell you about the work that we're doing looking at how the microbiome affects cancer chemotherapy. And so this is part of a broader topic in the lab where we're very interested in integrating microbiome research into pharmacogenomic and precision medicine approaches. And so, as you probably all appreciate, the traditional approach to pharmacogenomics ignores most of the genes in the human body. So, as we all appreciate, we're actually the majority microbial cells, and we also have at least an order of magnitude more microbial genes in our body than human genes. And over the years, we and others have started to describe kind of the general ways in which the microbiome impacts drug response. And we can kind of break this down into three major categories: direct metabolism of drugs, immune modulation, and transport and bioaccumulation. And just as a few examples, we've worked a lot on the Parkinson's drug levodopa, which can be metabolized by bacteria in the gut to dopamine and downstream metabolites. And this decreases the amount of drug available to the brain. In addition to that, I'll tell you a lot about 5-fluorouracil and kind of other types of cancer chemotherapy that can be metabolized by gut bacteria, taking an active compound and turning it into an inactive compound. There's also a really flourishing and busy area of research looking at how the microbiome affects cancer immunotherapy and other immune-related treatments. And interestingly, microbes can also kind of take up drugs and accumulate them in their cells. For example, it's related to neurotransmitters like serotonin, which can be taken up by certain members of the gut microbiota. And we're very interested in drug transporters like P-glycoprotein, which play a key role in both cancer drug resistance and intestinal drug absorption. But for today, I'm really going to focus on a couple recently published studies led by this amazing MSDP student in our lab, Kai Trepka, who's both an avid runner as well as a really hard worker in terms of the lab. And so, as a physician scientist, Kai is really motivated to understand how knowledge about the microbiome might improve medical care. I think we all appreciate both from popular media and personal experiences with our loved ones how really challenging chemotherapy is for patients. And this has, sparked a lot of controversy in terms of, what the best approaches are in terms of profiling both human genetic polymorphisms. For example, a recent lawsuit against Oregon Health, OHSU, in my home state, resulted in them paying $1 million due to the fact they hadn't genetic tested a cancer patient. And kind of the focus of this genetic test that was so controversial was a gene called DPYD. It's a very well-studied enzyme expressed in the liver and in other tissues in our body that takes 5-FU, a really common component of cancer chemotherapy, and inactivates it, forming this reduced metabolite DHFU. We got really excited by this area, both because of the potentially kind of off-target effects of cancer drugs on gut bacterial growth, but also the fact that there are now oral forms of these drugs that could interact with microbes before they ever get absorbed in the general circulation. In terms of the host gene, we know that about 5% of us, so probably at least one person in this meeting, have a known loss of function mutation in DPYD. And this can be really toxic if you're given high doses of 5-FU. But there's hundreds of other variants that have been discovered, and we don't fully understand kind of how they interact and what the consequences are. But in addition to that, there's kind of another layer of complexity that has really been unstudied for a long time, and that is that 5-FU inactivation can also occur in bacteria. And so, in our cells, these fluoropyrimidine drugs primarily act by inhibiting thymidylate synthase, which is a really important enzyme for replication of cells. And then, as I mentioned, they're cleared by this enzyme dihydropyrimidine dehydrogenase. In bacteria, it turns out that they are also sensitive to these drugs. We think through similar mechanisms. And just to make it a little bit more confusing, we call the bacterial enzyme pre-TA, but it has very similar activity to dihydropermanent dehydrogenase. And this was all described in an initial paper from our lab a couple of years ago now. But much of this work was in vitro and in mice, and so it really raised the question of whether or not there, how does this actually play out in patients, and is there actually a consequence in terms of either the efficacy or toxicity of drugs? And so we're really fortunate to meet Wes Kidder and the team of really talented clinical oncologists at UCSF. And about 6 years ago, they embarked on this longitudinal sampling study of colorectal cancer patients. The study design was relatively straightforward. We wanted to track what happened to the microbiome during multiple sequential cycles of chemotherapy. We ended up with a total of about 40 patients. Most of them were on capecitabine, this oral 5-FU as the primary drug, but some of which were also on alternative forms of oral chemotherapy, including capecitabine with immunotherapy, as well as a newer drug called TAS-102. We then collected stool at multiple baseline time points during cycle 1, and then at the beginning of each sequential cycle, and then finally at the end point. And so we're kind of grouping those into baseline, cycle 1, and trough time points. And so I'm not going to show you the compositional data, but we confirmed what we had previously seen, which is that these drugs do exert a clear effect on the structure of the microbiota. But I think perhaps more interestingly, there is also clear and consistent shifts in gene abundance. And so here we're just comparing baseline time points to the middle of cycle 1. And you can see along this component of the PCOA space, there is a kind of shift to the right. And this is significant using a multivariate analysis. Interestingly, when we look at the gene family or KEGG orthologous group level, we see a whole suite of genes that were enriched over time during cancer chemotherapy. And so we wondered what kind of groups of genes were more common in this list, and performed a pathway enrichment analysis. And so here you can see each of these pathways was significant, but the bolded ones are pathways that we know from kind of traditional uracil and 5-FU metabolism to be kind of directly related to how the drug might be processed in bacterial cells. And we were most interested in pyrimidine metabolism, which is directly related to the how 5-FU gets metabolized. And interestingly, when we looked at these KOs, you can see that each of these blue genes was significantly enriched over time during chemo. And you can kind of draw this whole pathway of 5-FU metabolism, including pre-TA, that key step for metabolizing 5-FU to its inactive downstream metabolite. We wanted to get some functional validation that these differences in gene abundance actually mattered, and so we incubated stool samples from these patients with 5-FU, and then measured how much was remaining. And here, as we predicted, you can see that as there are higher levels of pre-TA in the microbial communities, we see decreased level of drug remaining, indicating that there is a functional consequence for this enrichment in pre-TA over time. And so next we wanted to understand what the implications might be for toxicity. And so we established a mouse model of capecitabine toxicity, giving them 1,500 milligrams per kilogram, and did an initial experiment where we either kept their microbiota intact or gave them a cocktail of 4 antibiotics. And so here you can see that microbiota depletion really increases the amount of weight loss when given capecitabine. This is statistically significant both over time and at the day 5 endpoint. And we can also calculate a survival, do a survival analysis based on our humane criteria for euthanasia, and you can see that's also significant. We next wanted to do the inverse experiment. So here we start with germ-free mice and colonize them with a gut microbiota from a normal mouse. And again, consistent with the previous experiment, you can see that the germ-free mice are much more sensitive to the drug, with both the ex-germ-free or conventionalized mice, as well as mice that are colonized from birth, showing more stable body weights. This is also significant at the endpoint and by survival analysis. You can't really see it here, but both of the colonized groups are at the top. And so next we wanted to ask the question of whether or not we could rescue toxicity with a single gene, so with everything else remaining the same in the microbiota. And so we used a previously engineered knockout strain of E. coli where we removed the pre-TA operon, and then the strain expressing high levels of pre-TA. This is just to show that these strains did, colonize the gut of the mice to similar extents. But we do see a significant, although more subtle, difference in body weight over time. And this is also significant by day 8 in the survival analysis. And so next we wanted to do this in kind of a community level. And so we returned to our human samples and selected stool samples that were either low or high for pre-TA. We then colonized germ-free mice with those complex microbial communities and then gave them capecitabine. And so here you can see that these mice ended up with a clear difference in pre-TA abundance, with subset of them having below detectable levels of pre-TA by sequencing, and the others a range of different levels. When we dichotomize them into this high or low pre-TA status, you can see a clear difference in body weight that's significant by the end of the study, and we also see that in terms of other phenotypes in these mice, like pull and length. And so this really shows that differences in pre-TA abundance, both in alone and in the context of a complex community, can matter. And so finally, we wanted to understand whether or not this could actually be relevant to patients. And so we went back to our toxicity data, which we were able to retrieve from the medical record. About half of the patients had some form of toxicity throughout the study, which ranged from things like hand-foot syndrome, which are a common side effect of capecitabine, to other forms of GI toxicity, fatigue, and nausea, and others. And so just looking at all causes of toxicity, we do see that pre-TA levels were significantly lower at baseline for patients that went on to develop toxicity. And so for the rest of the talk, I want to focus on a more recent study where we asked whether or not there could be additional mechanisms through which the microbiome protects from chemotherapy, in addition to directly metabolizing the drugs. And, this study really came out of a really fortunate collaboration. As we were working on this study at UCSF, we became aware of another study ongoing at Maastricht University, and our collaborators, Marjolaine and Jolene, really kindly offered to work together to understand shifts in gene abundance in this cohort. And so they were able to collect 56 patients, also on capecitabine. Their study design is a little bit different, so they have a single baseline time point, a time point in the middle of cycle 3, and then an end of treatment. So we took all of these samples and subjected them to metagenomic sequencing as we had done previously for the US cohort. And so as I showed you before, we're very interested in gene and pathway level shifts. And so here you can see that at the pathway level, there again are many different microbial pathways that go up during chemotherapy. And we were really drawn by these points in the upper right of this volcano plot. And really strikingly, 7 out of 10 of those all came from related pathways involved in the biosynthesis of menaquinol, which is a reduced form of vitamin K2. But what are the genes and enzymes in these pathways? As we had seen previously for uracil metabolism, you can kind of draw the whole pathway of converting charismate to menaquinol through these enriched genes. Each KO in blue was significantly elevated during time in response to chemotherapy. But this does vary between patients. So while on average these genes go up, you can see that that only occurs in about 70% of the patients, with others actually showing either stable or decreasing levels of this pathway. And so this really raised the question of whether or not increased levels of production of vitamin K2 by gut bacteria could actually be protective or perhaps detrimental in terms of drug toxicity. And so first we wanted to understand what vitamin K2 does to the bacteria themselves. And so we used a transposon library that our collaborator Adam Deutschbauer had previously constructed in E. coli. We then grew this library in the presence or absence of three related compounds, so capecitabine and its downstream metabolites, GFCR and 5-FU. And then you can quantify the level of different barcodes in this library, which give you an indication of how sensitive a disruption of that gene, how disruption of a given gene impacts drug sensitivity. And so here you can see that insertions that go up indicate that the gene is actually detrimental, and insertions that go down indicate the gene is protective from the drug. And so I'll just focus for the sake of time on the protective genes. By the analysis we did, we actually found many, many genes with about over 100 that were consistent between the three drugs. And so again, we did a pathway enrichment analysis on these genes, and interestingly found that the top hit was quinine biosynthesis, including many of the genes that I showed you earlier that were enriched over time during chemotherapy. And with the most dramatic effects as expected by 5-FU, the more downstream active compound. And so this really suggests that the production of menaquinone by E. coli is protective. And so to more directly test this, we looked at growth of E. coli, either wild type strains or a strain with MenF, a key step in this pathway, deleted. We see that in the presence of 5-FU, the knockout is more sensitive, and that can be rescued by adding back menaquinone. And while we don't fully understand the mechanism, we had, based on the literature, some suggestions that it could be uracil dependent, and so we tested that. Here you can see in the absence of uracil, again, the knockout is more sensitive to the drug, but there's no difference when we supplement it in uracil. And so, you're probably wondering, this is interesting that bacteria are protected, but does this actually matter to patients? And so, fortunately, in the Netherlands study, they had really nicely curated side effect and toxicity data. And so we compared that all to the baseline levels of microbial pathways, and found three side effects were significant: peripheral sensory neuropathy, alopecia, and oral mucositis. And all of them have distinct pathways associated with them. For the sake of this talk, I want to focus on the neuropathy side effects. And here we found many different pathways that were depleted in patients that went on to experience neuropathy. And interestingly, at the top of this list were many of these same menaquinol biosynthesis pathways that we had seen previously. And so we next wanted to validate that menaquinone indeed was changing. And so this is just regraphing the DNA data. You can see that patients that had neuropathy had lower levels of these genes in their microbiome. But working with our collaborators at Tufts, we were able to quantify menaquinol, showing that one of its variants, MK28, was significantly associated with pathway abundance. And as expected, also really significantly depleted in patients that went on to experience neuropathy. And so this really suggests that the K2 is protective for mammalian cells. And so to more directly test this in vitro, we turned to a cell line, HEC293. We found that in the absence of 5-FU, there's no impact of menaquinol on cell growth. But as we add higher levels of 5-FU, we can see a rescue of cell growth in the presence of 5-FU. And then we also performed a mouse experiment where we tested the effect of menaquinone on our cat toxicity model. And so here we evaluated body weight, colon length, as well as a marker of neuropathy using a sensitivity to a hot plate. And while many of these phenotypes are unaffected by menaquinone, we did see a clear difference in this peripheral sensitivity phenotype, where menaquinone does seem to be rescuing these neuropathy-like phenotypes in mice. And so just to summarize what I showed you today, I've shown you that chemotherapy alters the metabolic activity of the human gut microbiome. We see increased levels of genes involved in the direct metabolism of chemotherapy drugs, as well as increased levels of pathways involved in production of vitamin K2. And both of those, we think, act to actually rescue the level of drug toxicity. And there's lots of interesting implications of this work where we might be able to leverage this knowledge now to choose probiotics or dietary supplements to mitigate the side effects of cancer chemotherapy, and that we're very interested in testing in intervention studies. But I think it's also kind of raised the importance of considering these interactions between different pathways within the microbiome and how changes in dietary intake of vitamins like K1 and 2 could impact how the microbiome interacts with our drugs. And so that's another area we're really interested in exploring in future studies. And both of these papers were recently published, and so I'd encourage you to check them out if you want the full story. They only showed a subset for the sake of time today. So with that, I really want to thank our funders. This work was really supported by the National Cancer Institute, as well as other sources. And in particular, Kai Trepka, the really talented graduate student who led all the work I told you about today. And then some former lab members, Ben, Van, and Peter, who really set the stage for this work in patient cohorts, due to their work in vitro and in mouse models. And so with that, I really thank you for your time and inviting me here, and would be happy to take any questions.